Welcome to the tutorial video on how to set up a ReactALL experiment in the software. This video will cover the following topics: starting a new ReactALL experiment, programming experiment steps, running the experiment and analyzing results. To begin, open the crystallization system software by double-clicking on the icon. The available ReactALL system will automatically connect. For detailed guidance on navigating the software, refer to the tutorial video Introduction to the ReactALL software. The main screen displays the Experiments tab. The Experiment screen further consists of different tabs. In the File tab, you can open a previously saved experiment or start a new ReactALL experiment. Below, you can see a list of open experiments, as well as recent experiments. This offers easy access to the experiment files. The status of each open experiment is indicated by a color: idle, running, pause or prompt, finished but not saved, finished and saved, error, analyzed but not saved, and scheduled experiment. To start a new experiment, click on New ReactALL Experiment, which will open the Details tab. Fill in the details of the experiment, such as the experiment name, username, and description. Enable the available view options. Some options are grayed out and cannot be enabled. Next, select the type of cap. The only available stirrer option for ReactALL is the overhead stirrer. Enable the reactor reflux to maintain the reflux temperature, which is located at the top of the reactor vial. The reflux temperature helps cool the evaporating solution, preventing solvent loss. Next, enable or disable the wait for reflux option. When activated, the system will delay the start of the experiment until the desired reflux temperature is reached. The sample tube filter function enables the best sample sequence settings when the filter for it is used for filtering the solids out from the sample. Sequence settings, if needed, change the duration and pressure of different sampling sequence steps based on the type of quench, dilute, and rinse solvents, or use the default settings. To learn more about sampling sequence settings, check out the ReactALL Sampling Sequence Settings document, linked below this video. Fill in the vial details: compound, solvent, concentration, supersaturation, and choose the type of stirrer used for the experiment. The helix stirrer is recommended for most experiments, while the impeller stirrer is used when a Raman probe is integrated with ReactALL. Next, proceed to the Program tab. Here, you can program the experiment by dragging the desired step from the actions list. The available actions are: ramp, increase or decrease the temperature using a linear or quadratic ramp; hold, maintain a constant temperature for a specified duration; prompt, keep the same experiment conditions until you click Continue; tune, calibrate the transmissivity to 100% at a clear solution; sample, take a sample from the reactor, which is followed by quench and dilute; rinse, clean the reactor tubing and needle with rinse liquid; purge, blow nitrogen into the reactor vial to make an inert environment for reaction; blank sample, take a sample using only quench and dilute liquids, without collecting an actual sample. This can be added between actual samples to check for contamination. The program starts at 20 degrees Celsius by default, but this can be adjusted on the right side settings. At this step, it is recommended to specify image interval, stirring speed, and reflux temperature, as these specifications will then be applied to all subsequent steps, avoiding the need to re-enter them each time. To program an experiment, drag and drop the required action in the program steps. A tile will be red if the input is not complete or incorrect. For each step, fill the details on the right. For example, for linear ramp, insert the end temperature, heating rate, image interval, stirring speed and reflux temperature. Similarly, add more steps as needed. When sampling is required during the experiment, add a sample step, followed by a rinse step. It is always recommended to add a rinse step after each sample step. A purge step is automatically included when the first sample step is added. Add a hold step if necessary. The time required to execute each step is displayed on the respective tile. Generally, it takes 3 minutes and 40 seconds to complete a sample step, and 1 minute and 45 seconds for a rinse step. Each row is numbered sequentially, 1, 2, 3, etc., and you can repeat each row by increasing the group repeat to the desired number of times. The total time for each row is also shown. Similarly, add more rows if more steps are required. Steps can be added below or in between the rows. Some options are available at the top of the program. These are: import from the library, export to the library, import from a CSV file, export to a CSV file, delete to remove a program step or group from the program, and duplicate the selected program step or group. The steps programmed will be shown in the program chart below with different step numbers. After programming the experiment, proceed to the next tab called Run and Results. Select the reactor where the smart cap has been placed to perform the experiment. Before starting the experiment, in the Sampling tab check that the vials are scanned and that a sufficient number of empty vials are available in the respective reactor row. If the number of detected vials is fewer than the programmed sampling steps, the software will display an error. Once vial detection is confirmed, you can start the experiment by clicking the play button or schedule the start time for the experiment. The start of the experiment is highlighted in the software and reflected by a blue light above the selected reactor. You can pause or stop the programmed experiment by clicking on the respective buttons. You can eject the experiment from the assigned reactor only after the experiment is complete. The graph shows the live trend of the experiment parameters like reactor and target temperature, reflux temperature, target reflux temperature, stirring speed, transmissivity, samples and program steps. You can select or deselect the parameter to be shown on the graph by clicking on the respective label. The program steps display the program row, group number, and the repetition count for each group. The purge, sampling, and rinse steps are shown as blue shade. And the blue line is point of actual sample taken. The sample number is displayed at the top. The samples collected can also be seen in the Sampling tab. The successfully sampled vials are represented in green. Click on the sampling region to know the time of sampling. The second click will show the time between 2 points. You can zoom in by dragging the cursor along the axis. There are several options available by right-clicking on the graph, including Reset Zoom, Clear Selected Item, Copy Image to Clipboard, Select Data, Crosshair, and Add Comment. You can add a comment to note any specific conditions that occurred at a particular time. This comment will be included in the exported CSV file. Below the graph, various experiment parameters are displayed. To see the data at any specific time, enable Select Data and drag the line. The captured images are shown below the graph. You can navigate between images by clicking the arrows, or by dragging the crosshair along the graph. To adjust the camera settings, click the Settings button and configure the optimal image capturing settings for your sample. To add any step during the experiment, pause the experiment and go to the Program tab to add the required steps. The yellow color of the tile shows the current step. Drag and drop the required step and fill in the details. Make sure no sampling step is taking place during the pause, which can be checked in the Sampling tab and the sampling state. Then start the experiment again. When the experiment is complete, a green tick will appear in the software, and the reactor color will turn green. Click on Export to export the experiment data. You can export the whole experiment or the specific duration of the experiment by filling in the time interval. The experiment can be exported as results, which is the CSV file, images, or movie. At the end of the experiment or even during the experiment, you can go to the Analyze tab to analyze the data. The data can be analyzed as image analyses and transition points. The performed experiment will be available in the File tab until it is deleted by clicking on the discard sign. To save an experiment for future analyses, click on Save and Close. To copy the experiment program to another reactor, click on Copy. If you have any questions, please reach out to us.